talk is going to be given by Ajat Azimi, and it's titled Representation of Spatial Frequencies in Neural Populations of Human Temporal Cortex.
Okay. Hello, everyone. Thanks for accepting our invitation to this keynote. I'm Hojat Azimi, a master's degree student in artificial intelligence at the University of Tehran. The title of my talk is Representation of Spatial Frequencies in Neural Populations of Human Temporal Cortex. Actually, this is the result of our latest work, which I'm going to talk about. Let me start my presentation with a little game. Here, all of you must probably see a little red dot in the center of your screen. I want you all to just look at it. Please just focus and look at it. Don't blink or anything. Okay, 1, 2. Please don't look anywhere else, just look at the red dot. 1, 2, 3. Okay, as you must have seen, for a short period of time, you must have seen a picture. But the question is, what did you see? Could you describe it? How was it? Was it animate or inanimate? Like the picture on the right. Was it more similar to the chair or to the dog or to the apple? The next question is, did it contain any human faces like the picture on the right? And finally, was it a blur photo or a sharp one? Maybe you can answer these questions or no. But we have these questions in our mind, whether our brains are our brains capable of answering these questions or not. To answer these questions, we designed an experiment. Our experiment contained 155 grayscale images, including 73 animate images and 82 inanimate images. We showed these images, actually you can see our stimuli set on the right side of the screen. We showed these images to one subject and recorded 6 signals. Our subject was actually a pharmacologically intractable epileptic patient who had 50 electrodes in his temporal cortex. We had selected 5 areas in the temporal cortex, including right hippocampus, right and left supplementary motor area, right pre-supplementary motor area, and the right anterior cingulate gyrus. We had 10 electrodes in each of those 10 areas. After that, we set our paradigm for our experiment, which was really close to the little game that we did together in the beginning of the presentation. Each trial started with 100 milliseconds of fixation. After that, we showed the stimulus for about 200 milliseconds to the subject. And after that, the subject had to choose whether he has seen a face or a non-face picture, following a 500 milliseconds of rest. Okay, after actually recording these signals, we wanted to see if our signals have any information about the questions we had in mind. We started by visualizing the signals in the form of ERPs. Here you can see the ERPs of the 3 categories of signals based on their spatial frequencies on the right side. Here we have plotted only the signals from right hippocampus. But let's have a closer view. Let's compare each categories. Actually, let's compare them in groups of 2. Here we can see that by comparing the categories 2 by 2, there are some points in each trial that there are significant differences between each of the 2 categories. This means somehow that maybe actually a different representation of these type of images in the brain. To discuss this more, we actually trained some classifiers. Here you can see the performance, the accuracy of our SVM classifier through time. We did the classification 2 times. Once for a face versus non-face classification that you see here. Here we have the whole population of neurons on the left side of the screen, and on the right side of the screen, we have trained our SVM by the use of only the right hippocampus. We can see that as the time goes through, our classification accuracy goes high, which means the perception of the image category actually. After that, we again repeated this experiment, but this time to classify between high spatial frequency versus low spatial frequency. Here again, we can see in the form of bar charts with standard error drawn in red lines, that our accuracy goes high, and again, there's a significant information behind the signals that we have recorded. After that, we wanted to see how distinguishable our signals are from each other through time based on their categories. To do that, we did a separability index analysis. Let me have a short review of separability index. So in order to calculate separability index, we first calculate separability between each class, between actually the subjects, the samples of each class, which we call here actually as indexed I. And the total within class scatter is called as index W. We then find the scatter of between the classes, which is called as index B. Finally, our separability index is calculated by this formulation. Okay, now let's see how our SI changes through time. Here we can see how our temporal dynamics of SI is. Here the 0 indicates when we are showing the stimulus to the subject. As you see, the signals start to separate from each other through time right after we have shown the stimulus to the subject, and it has a nice peak here. So we can say that there is almost information in the form of our signals. Actually, that's it because of the short time that we had. But we have done much more analysis that I like to show to you, but I don't have time. So if you want to know more, I'd be grateful to show them to you. Feel free to contact me. Thank you.
So thank you. That was a really great presentation, really clear. We actually, so if anyone has questions, they should post them in the Q&A. You do have time to share the results there if you would like to, just to generate more questions. Otherwise, we can open the floor for discussion, whichever you'd prefer.
I believe it is better if you let them ask their questions.
Great.